You're most welcome at Medical Sciences Made Easy, and we are always privileged to have you on this channel where we learn a lot. And today's session we want to look at the structure of nucleic acids, what we call DNA and RNA. After looking at how they look like, what they are composed of, we shall also focus on the differences between RNA and DNA. So let us get started. And we always remind you to subscribe, share your comments, and like the video. So nucleic acids, which we know as DNA and RNA, to begin with, DNA is made up of nucleoproteins. It is made up of nucleoproteins, meaning we have the nucleic acid bound to the proteins, and these proteins are known as protamines and histones. And this DNA is found within the nucleus of the cell. It is purely found in the nucleus, that is greater than 90% found in the nucleus, and small percentage found in the mitochondria. Less than 10% of the DNA is found in the mitochondria, but the majority are found in the nucleus. So this is DNA made up of nucleoprotein. So a combination of protein and the nucleic acid forms the nucleoprotein, which is protamine and histone. And we are seeing the site where it is located is in the DNA. Then to the smaller extent, we find it in the mitochondria. That is the DNA. Whereas the RNA we shall talk about for it, it is located in the cytoplasm. It is majorly found in the cytoplasm. That is greater than 90% found in the cytoplasm, then less than 10% found in the nucleolus. Less than 10% is found in the nucleolus. So DNA, which we want to discuss, and RNA, especially DNA, is made up of, we're going to tell you that DNA is made up of nucleoproteins, and these nucleoproteins, we are going to see it is made up of polymers of nucleotide. So that's why we call it a polynucleotide molecule. These are macromolecules which are polynucleotide, made up of nucleotides, many polymers of nucleotides, and these nucleotides are subdivided. They are composed of two elements. That is a phosphate group. They are made up of a phosphate group. They are made up of nitrogen bases, and they are made up of, we can start with pentose sugar. They are made up of pentose sugar, and lastly they are made up of nitrogen bases, whereby the phosphate, it has a phosphate, then it has the pentose sugar, whereby in DNA the pentose sugar is deoxyribose, whereas in RNA the pentose sugar is ribose. So in the DNA we call it deoxyribose because if this is carbon number 1, at carbon number 2, the DNA lacks the hydroxyl group. It is removed. It has only the hydrogen group, which is different from RNA, whereby they possess the hydroxyl group at carbon number 2. Then the nitrogen bases include, whereby in DNA we shall see, in DNA we have thiamine, and thiamine is always complementary to adenine, whereas cytosine is always complementary to guanine. Whereby thiamine and adenine, they are always having a double bond. These ones are connected by triple bond. Then in RNA, we shall see the nitrogen bases. In RNA, we don't have thymine. Instead of thymine, we have uracil. And uracil is always complementary to adenine, then cytosine by a triple bond hydrogen bond with guanine, whereby these nitrogen bases are divided into purines and pyrimidines. We categorize them into purines and pyrimidines. And purines, we only have two, that is guanine and cytosine. These are purines. That is guanine and adenine are purines. Then pyrimidines, we have cytosine, we have uracil, we have thiamine. So these are pyrimidines and purines. So that's how we categorize these nitrogen bases in nucleic acids. But we have ended up saying that in DNA it is thiamine. Thiamine is peculiar for DNA, so we cannot find thiamine in RNA. And we cannot also find uracil in DNA. So in DNA we have thiamine, in RNA we have uracil as nitrogen bases. So after seeing this introductory part, now we can look at the structure of DNA in details after knowing where it is located, knowing what it is and what it is composed of, which is made up of many polymers. And these nucleotide bases are always joined together by phosphodiester bond. We are going to see that these multiple nucleotides are joined together by phosphodiester bond. So phosphodiester bond is the one that joins these nucleotide bases. So we're going to see that if this is a chain of nucleotide base, and even this is a chain of nucleotide base, and as we are looking at the structure of DNA, this is the structure of DNA, we are going to see that this nucleotide base made up of a phosphate group, a pentose sugar, and we said in the DNA the pentose sugar is known as deoxyribose, and then they have also nucleotide bases at position number 1. And we are going to see complementary base pairing, whereby this one will be at 3' end versus 5' end. 3' end versus 5' end. So this one is going to move from 3' end to 5' end, and this one is going to be moving from 5' end, 5' end. So this one is going to be moving from, so they are antiparallel. So 5' end, then this is also going to be moving to 3' end. So this is what we say that the DNA is antiparallel. This one is from 3' end to 5' end. So according to the structure of DNA, which we say that DNA is a helical structure, it is a double helix. It's a double helical structure. It has double helix made up of 2 nucleotide chains, and these 2 nucleotides, they are joined together by nitrogen bases. They are joined together by nitrogen bases, whereby if this is thiamine, this is adenine. If this is cytosine, this is guanine. If this is adenine, this is thiamine. This one is joined together by double bond, hydrogen bond, and this one is joined together by triple covalent hydrogen bond. So the 2 strands, if this is from 5' end to 3' end, this one starts from 5' end. So for us, according to our discussion, we are beginning from 3' end to 5' end, then from 3' end to 5 prime end. So they are anti-parallel. That these double helix in the DNA, they are anti-parallel. They are anti-parallel. Why? Because one begins from 3 prime end to 5 prime end, and another one moves from 3 prime end to 5 prime end. And according to Crick and Watson, this is according to Crick and Watson description. And this Crick and Watson, they tell us that DNA is right-handed. That the majority of DNA is right-handed. That during coiling, they coil on the right-handed side. That is especially DNA type A, type B. DNA type B, DNA type B, which is, and sometimes A, which is right-handed, whereas other types like A and Z, they are left-handed. So we are going to see that this is antiparallel, and we are saying that they are joined together by phosphodiester bond, in that the hydroxyl group at carbon number 3 of the pentose sugar interacts with the phosphate group at carbon number 5. This is carbon number 4, this is carbon number 5. So we shall see them interacting at carbon number 5, leading to the removal of the hydrogen group. And this one also comes and interacts with the phosphate group at carbon number 5 to form another phosphodiester bond. And another one comes at carbon number 3 and interacts with the phosphate group at carbon number 5. That's why we are calling it 3 to 5 prime end direction, because the hydroxyl group at carbon number 3 is joining together with the phosphate group at carbon number 5. And these are the ones we are calling phosphodiester bond. So this is what we call phosphodiester bond. Then we shall also see that these ones which are beginning from, it is also still the hydroxyl group at carbon number 3. This is carbon number 1, number 2, number 3. Is going to interact still with the phosphate group at carbon number 5. So we're going to see the formation of a phosphodiester bond, another formation of a phosphodiester bond, another formation of from 3 to 5 prime end. So that's why we say it is double-stranded. And these are the 2 strands of nucleotide bases joined together by phosphodiester bond. This is a phosphodiester bond. So they are joined together by nucleotide bases. And we said, in case at this point we have cytosine. Cytosine is in complementary base pairing. Cytosine is complementary to guanine, and adenine is complementary to thymine. And we said cytosine and guanine are joined together by covalent bonds, and they are triple. It has triple hydrogen bonds. Whereas if it is adenine, is complementary to thymine, and they are joined together by double hydrogen bond. So this one is joined together by a double hydrogen bond. Then this is triple hydrogen bond. So if another here we have adenine, we have thymine. Thymine is complementary to adenine by two, or what we call double hydrogen bonds. And if this is guanine nucleotide base, it will be complementary to cytosine by a triple hydrogen bond. So we are saying that the 2 strands are joined together by complementary base pairing, whereby cytosine guanine with a triple hydrogen bond, then adenine thymine with a double hydrogen bond. So this is the structure of DNA. This is the molecular, this is more detailed, and this is what. And we find them, they coil all over. And the different types of DNA we have looked at is we can talk about DNA type A, type B, and type Z. But the most abundant is DNA type B. And we are saying that these strands, which are joined together by the nitrogen bases and these nucleotide chains by the phosphodiester bond, they coil over the other in a right-handed direction, that is especially in DNA type B. And this coiling, right-handed coiling of the two strands which are antiparallel, joined together by the nucleotide bases, and this one is the phosphate or the phosphate backbone. We say them that one turn, if one strand of DNA coils and makes two turns, joined together by 34A, then they have what we call the major and the minor groove. This is the minor groove, and this is the major groove. And these grooves, they join together by a 3.4A. So this is the structure, all of them having nucleotide following complementary base pairing, whereby cytosine with guanine, adenine with thymine. Then here they have no uracil, adenine with thymine. So complementary base pairing still holds cytosine, guanine, then adenine, thymine, whereby here we have triple, we have triple still, and here we have double. So they are joined together by these are nucleotide bases. So this is basically the structure of DNA which we need to talk about. So after understanding the structure of DNA in this session, another thing we need to look at is RNA. And for RNA, which is ribonucleic acid, these ones, for them, they are single-stranded. It is single-stranded. It has one polynucleotide chain. So if it is an RNA, it has only one polynucleotide chain joined together by phosphodiester bond. So it is a single-stranded, and we said it is found majorly in the cytoplasm, then to smaller extent in the nucleolus. And the types, it has different types of RNA, whereby we have types like messenger RNA, we have transfer RNA, and we have ribosomal RNA, and small nuclear ribonucleic acid. So these are the major 4 types of RNA, but the major ones are first 3. Then this one, which is found in the nucleolus, is also part. And to know that the major function of messenger RNA, for it, if this is the cell, and inside we have the nucleus, we are saying that the smaller ribosomal subunit are found in the nucleolus. Then if this is the DNA in the nucleus, during central dogma, that is replication, transcription, and translation, first this DNA in the nucleus, it replicates, and after separating the double strands, it undergoes transcription to form an mRNA. And this mRNA, it is small, so it passes through the nuclear pore and goes to the cytoplasm, where there are ribosomes. This is the ribosome. And this mRNA, which has moved into the cytoplasm, it goes into the machinery which we call ribosomes, where translation occurs to form proteins. So the major importance of messenger RNA is the one that transports genetic information. It is the one that transports genetic information from the DNA, mother DNA. Then it is taken to the cytosol, where ribosomes are located, and they act as a machinery for translating the genetic information from the big DNA and it translates it into a predetermined protein. That is the function of the messenger. Messengers are always sent to send a message. So it sends the message from DNA and it is translated into a protein. And we are going to see another the importance of transfer RNA. For it is acting as an adapter. Because these mRNA, they always have codons, whereas the transfer RNA carries anticodons. It carries anticodons together with the predetermined amino acid charged with an amino acid. It is always charged with amino acid, whereby this transfer RNA, it brings the complementary codons, and these codons specify for an amino acid to be added on the polypeptide chain. So that's why we are calling them that they act as adapters, whereby they come and bring the complementary anticodons to codons on the mRNA, specifying the amino acid to be added during translation. That is the major importance of transfer RNA. Then ribosomal RNAs, these are the ones that are found within the ribosomes, responsible or whereby they are acting as machinery for protein synthesis, and the small ribosomal subunit, they are the one responsible for cutting. So these ones, we know that when mRNA is produced into the cytoplasm, it needs to undergo post-transcriptional modification, whereby we need to remove the introns and we join the exons, which are coding. So what cuts out the introns and join the exons is what we call small. So this one does the role of splicing. Splicing and excising. The coding and the non, it removes the non-coding and join together the coding, which are the exons. So this is smaller ribosomal nucleic acid. So this is basically what we can talk about on the RNA, which is single-stranded, found major in the cytoplasm and smaller extent to the nucleolus, and having major 4 types, and we have seen even this function of each type of RNA. So as we are concluding in this session, whereby we have been looking at the different structures, that is the RNA and DNA, and maybe to tell you about the structure of transfer RNA. Transfer RNA, it has what we call 3 arms, whereby these 3 arms, whereby if this is a 3' end, so it becomes a 5' end, and these are the ones that transport. This is the loop that has the anticodons, which specify for the type of amino acid to be added, and then we always see them being charged to carry the certain amino acid. So this is what we call a transfer RNA. So this is what we can talk about, and as we are concluding, we are going to look at the differences between DNA and RNA. Differences between DNA and RNA. So we want to look at the differences. And this is commonly asked in exams, so you always need to know the differences between DNA and RNA. So this side we shall have DNA, and this side we shall have RNA. And we said that DNA is double-stranded. It is a double helix or double-stranded. It's double-stranded, whereas RNA is single-stranded. And we have said in DNA, the nucleotide bases, we have thymine, we have thymine, we have guanine, we have cytosine, and adenine. Whereas the nucleotide bases, these are nucleotide bases, these are nitrogen bases. Whereas here, the nitrogen bases in RNA, we have Instead of thiamine, we have uracil, we have guanine, we have cytosine, and we have adenine. So in short, we are saying that DNA has thiamine, whereas RNA, they have uracil. So this one has uracil and lacks thiamine, and this one lacks uracil, has thiamine, which you abbreviate as T. This one we abbreviate it as U. And we saw that these ones are majorly found in the nucleus, and at smaller extent and small in the mitochondria, whereas these ones are majorly found in the cytoplasm and smaller extent we saw them in the nucleolus. We saw the different types of DNA as type DNA type A, type B, and type Z, whereas the types in RNA we have seen messenger RNA, we have seen transfer RNA, we have seen small ribosomal RNA, and ribosomal RNA. So these are the different types we have seen in all of them. So in summary, we are going to see that in DNA, adenine is complementary to thiamine. This is very important. Whereas in RNA, adenine is complementary to uracil. This is very important to emphasize. Very important. So these are some of the differences between DNA and RNA. And as we conclude, we also have a rule which applies only to DNA, whereby DNA obeys what we call Chagaff's rule. That DNA obeys Chagaff's rule, whereby Chagaff's rule says guanine plus adenine is the same as cytosine plus thiamine. So all these ones must be equal. So the ratio, if I get the ratio of adenine plus guanine and I divide it cytosine with, I add it with thiamine plus cytosine, the ratio should be 1. So if I deny you any figure and I give you this of guanine should be the same number as cytosine, and adenine should be the same number as thiamine. That is what it means, because they are always complementary. And if I have, so after knowing that guanine must be the same as cytosine and adenine should be the same as thiamine, that's why the ratio according to Chagaff's rule is 1. Thank you for listening. Until the end, may the good Lord bless you.